Okay, we're going to go ahead and get started again. Now we're behind. Okay, so yesterday we had our scientific meeting across the way at Cincinnati Children's. Again, thank you so much to Dr. Huppert for hosting that and for pulling together what can only be like the most impressive group of folks working on Alagille syndrome or interested in Alagille syndrome. It was amazing. Thank you so much for doing that. Dr. Huppert is going to kind of go through an overview of what they discussed yesterday at the scientific meeting. So if you want to come on up. Thank you.
Sherry, what time are we supposed to go to so I can get us back on time? Oh, it's 10:42, so do you want me to take more than that?
Be quick.
Now we're really behind.
I can be quick. Can I do 5 minutes?
Oh, no, you can do it. You can do it.
All right.
As much time as you need.
Well, I don't want to, whoa, I'm a loudspeaker. Okay. So I definitely want to thank all of the clinicians and researchers and families that showed up yesterday. It was a great day. We had great interactions, I think. A good number of attendees. We had, I think, over 80 people that were there. We had 19 phenomenal speakers that walked us through quite a huge ground of area, starting from clinical outcomes and then moving all the way into basic science and kind of where we are. So we started with where we were, and I'm going to put David Piccoli on the spot to stand up. Back there, he let us, you can stand up and wave, although everybody's seen you already today. But so David kicked us off with where we were and where we are with Alagille syndrome, and kind of gave us an idea of where we need to go when we're thinking about doing research, some of the clinical areas that we need to be concerned about. And then Benita, which you've heard talk already today, really put us into this idea that there is continued importance of collecting clinical data, especially as Alagille patients. Most of them are seen in the clinic, as you saw in the survey, when they're very young, but what happens as they become adults. And Benita's starting to collect information in that area. And then we had an amazing family experience talk from Tara, and she shared her story with her Alagille warrior, Cord, her son, and that was very impactful for all the researchers to see. And then we dug into the mutations in the genes JAGED1 and Notch2, which you've heard about, and many of you know some of your genetic diagnosis that is associated with Alagille syndrome. And just to make this pathway is really, JAGED is the ligand or the signaling that's in one cell, and Notch is the receptor in the other cells, and it's a communication pathway. It's how information is transferred from one cell to another, and that's how cells make a decision in your body. And that's why many things go wrong and are observed in Alagille patients, because almost every time a cell makes a decision in the body, it uses the Notch pathway, JAGED and Notch, to make that decision. So Nancy led us down, telling us about some mutations that's found in JAGED and Notch, and some of those mutations, what results in that is that the ligand, JAGED, doesn't make it to the surface, so it's not able to interact and see the receptor. And so that explains some of the, potentially some of the defects that we see. And then Vince Luca, where is he? Back there in the corner. Vince talked about interactions and the structure of both the ligand and the receptor when they complex together, and told us that some of those mutations that are found in patients, how they may influence signaling activity because of the strength of that interaction between the ligand and receptor. And it's really important, I think, one other point, because this is a signal that's on the surface of one cell communicating with a receptor on another cell, and then when that interaction happens, the receptor goes to the nucleus and activates genes. And so there's no amplification event. So it really is strengths when we're talking about ligand and receptor that influence all the output and the phenotypes. We had, and Bob is back there in the corner, and he talked about modifications that happen on the ligand and on the receptor that can either activate or inhibit, basically increasing or decreasing signaling levels that also will give you that different pleiotropic phenotypes that happen in different cells and different organs of the body. And let me move on. Okay, and then we had a very special speaker, Anne Jotel, that came to us from Paris, France, from the INSERM, where she leads a big group on pathogenesis of small vessel diseases in the brain. And she gave us some interesting ideas about how jagged and notch may be regulating the integrity of vessels. And Anne's sitting right here in the front with us still here today. And then Jan is sitting here in the middle. He's at the University of Illinois, and he also talked about angiogenesis and interactions between the ligand and receptors and how you build vessels. And Brenda Lilly at Nationwide Children's also followed up on that idea, how you get cells that are part of a vessel, because there's multiple cells that make up a vessel or the integrity of a vessel, how they can be impacted by lessening the level of notch activity. And then we went into an area of organogenesis. So we mostly talk about the liver disease when we think about Alagille patients, but I wanted to go into some areas that we don't necessarily talk about. So Lindsay from Cincinnati Children's, she talked about facial structure, developing the bones and the facial patterning, both in the face and in the middle ear. And then Kathy, who's sitting over here, talked about bone density and the DEXA scans that I think have already been mentioned today. And then another Cincinnati Children's colleague of mine, Giuseppe Park, talked about kidney development. So how does notch regulate the different cells of the kidney and the progenitor cells? Okay. And then we had a biomedical engineer from Wake Forest, Shay Sokar, that talked about liver organoids to study both development and do drug discovery screening in. Emma is over here. Emma talked about her mouse model, the jagged mouse model, and three-dimensional structure of both the vascular system and the biliary system in those models, and the structure and how they may impact each other. Hamid is in the back there. He talked about about dosage of the notch pathway and how other genes and mutations in other genes may modify the phenotype of Alagille, may impacting the severity and the spectrum of the disease. So he talked about another gene, SOX9, that impacts and makes a jagged heterozygous mouse worse. Is Duke still here? He's right there. So Duke talked about his zebrafish model and looking at liver bile duct regeneration, and also a screening compound that he's found that looks like it elevates notch activity. Is Alison here today? Okay, Alison was from the University of Penn, and she's a postdoc with Ben Stanger, and she is working on the regulation of the chromatin that allows accessibility of genes to be transcribed in cells in the liver that are reprogramming from hepatocytes to cholangiocytes to build bile ducts. And then we wrapped up the afternoon. I hope I didn't miss anyone. I think I got everyone. We wrapped up with Holger in the back, one of my colleagues, and he spoke about some of our work with inducing transdifferentiation in models of Alagille syndrome, and he's shown that you can transplant in normal hepatocytes and they will convert into cholangiocytes, and also some overexpression of jagged in hepatocytes to improve and induce transdifferentiation of hepatocytes into cholangiocytes to build structure. And then we ended with Taka Takebe from Cincinnati Children's that is working on building organoids for different organs in the body, and mostly liver he's focused on, so that we can do drug screening and have multiple cells within that liver organoid so we can see the impact of other cells and toxicity within it. And lastly, he's working on building a model that shows you the full connection that has the liver into the extrahepatic bile duct and into the duodenum, so we can study the interactions between those types in cells that were derived from human and do that in culture. So I think I summarized, hopefully. If you have more questions, as I pointed out, everyone, most of the speakers are still here, so they'll be very receptive to talk to you and talk about their research.
Thank you so much. As she mentioned, a lot of the researchers and scientists are still here, and we want to see more of that. We want to see our families talking to our scientists, talking to our clinicians. We just want to get a good mix at each table later today when we get into our working groups. Right now, we are scheduled to have investigator perspectives, which is a pass the mic, like we did a little bit earlier, but give our investigators a chance to let us know, number one, what they need to know from us, and then one of the things that we asked them on their survey is, what do you feel like is misunderstood about ALGS research? And we had some great answers, and so I would like to hear from some of them. It seemed like the patients asking questions or the families asking questions was really impactful. So maybe let's go through some investigator perspective, and then we'll move back into the same format that we were on a little bit earlier. And now we do have to stay on schedule because she's going to kick me out because symposium is starting one way or another. So we're going to go until 11:30, and we'll get as much done as we can. So you have a mic. There's another mic. Do you have both? Oh, right there. Okay. So if you want to just raise your hand, if you are an investigator, a scientist, an industry professional, someone working in the space, and want to answer one of those questions, please raise your hand, introduce yourself, and then we'll just keep on passing the mic around.
Okay, so I'm Jan Kidieski, and I study how jagged might promote the development of blood vessels. Now, I've spent a lot of time trying to inhibit jagged, I'm sorry to say, because it also has some implications in how tumors evolve. But what I have learned is that we can manipulate the protein and its activity. And I think I've been inspired from the stories and from hearing not only patients and hearing about warriors, but other clinicians, that maybe there's a need to explore whether we can tweak this protein in a way that might help amplify the one good jagged that all the warriors have. And so that's something that I'm taking from this meeting and thinking about as I go back to my home institution. So thank you all for your perspectives.
Great. Thank you. Just raise your hand, and we'll come to you. Right over there.
Holger Wilmbring from UCSF. One of the questions from the survey was, how long is it going to take reprogramming of the PAL sites into, or cells in the liver like a PAL sites, into cholangiocytes and bile ducts to the clinic? And Stacy and I were particularly or specifically working on that, and I want to follow up on what was just mentioned. So we do believe that it is feasible, and it will probably not take a long time to make this happen, in particular in mouse models, to an extent that is very, very effective. But the one thing we're then faced with is the safety aspect of this approach, and as it was just hinted at, notch signaling is definitely a pathway that can drive cancer and also fibrosis in the liver, and so it's something we have to be very mindful of when we're trying to translate these approaches, that we not make things worse actually with in the process of trying to build a biliary system from scratch, and so we're trying to be as specific and as targeted about this as possible. And some of the approaches that were highlighted yesterday, actually, I feel have potential in that way, that are very specific. They can be more specifically controlled with regards to the time that the activation is being induced, and also hopefully with regards to the location where it is induced. So these are the kind of things that make translation more time-consuming, more complicated, but then hopefully also more successful at every level.
Thank you.
Yeah, my name is Hamid Jafari. I work at Baylor College of Medicine in Houston, and I wanted to share briefly with you my experience as a scientist, as my work kind of evolved from pure basic science to work that is related to human rare disease. So currently my lab works on 2 different rare diseases. One of them is Alagille. One of them is much rarer, like 50 patients in the world. And 6 or 7 years ago, our work did not have any direct connection to any human diseases. We were working on pathways that we knew are important for human biology, but it wasn't relevant to any diseases. And over the last several years, I've been to several meetings for Alagille and also for NGLI1, and I wanted to tell you that interaction of scientists with patients and patient families can also be transformative for the way we view our own work, we view even, kind of the whole philosophy of why we're doing this. So don't underestimate the meaning that you can give to our lives. So this is a bi-directional avenue, in the sense that I've heard this several times from patient families that, whenever you're down, whenever your experiments are not working, know that we're hoping that you guys find something. You bring meaning to what we do, and for that I think I might be able to speak for other scientists in the room. We are thankful to you as well. So this is a bi-directional work. I wanted to let you know that sharing your frustrations with us is really impactful. So when I go back to my lab, I'm going to tell my lab members that, this is what we heard from Alagille parents. Guys, if you come work on a weekend, you're not just doing this for me or for yourself. So please. So there are a lot of things that is way beyond my ability or the clinician's ability, when it comes to insurance, when it comes to regulatory processes. But I would like to reassure you that we are doing what we can, and we also share the hopes and worries similar to you that, one day what we're doing might be able to, have an impact on the lives of your children.
Thank you, Ahmed.
Thank you.
Thank you.
Can I just say that's a really important aspect as far as our organization is concerned also, because if we don't hear from families and families don't participate with us, we can't give scientists what they need, and we can't facilitate these kind of meetings if we don't have family participation. So it's so important that, as an organization, that we connect with the families and families trust that we're facilitating these kind of things as well.
So I guess this is for other scientists. I'm Duke Dong. I'm one of the researchers. Going through my scientific career, I think one of the biggest things I've learned in getting science to work is to really avoid the naysayers, people who say things won't work. I've had PIs told me past projects, things will not work, things will not work. When you, as another PI, review another person's grant and so on, keep in mind that it's really easy to say, oh, that won't work, but you also have to consider approaching it in a more positive way. How can you make it work? How can you overcome that obstacle? I think that's really the mentality to take when we approach science. I've had my past PIs say things will never work, would laugh at me, and in the end, I got it to work. I bring this up because as soon as I bring up the possibility of using a notch agonist, I've been told by multiple people already that, it's also implicated in cancer. Actually, if you read papers out there, there are people who are trying to use that notch agonist also to prevent cancer. And keep in mind also, I'm just using it as an example, cancer, keep in mind, is a mutation-based disease. If we do use an agonist for this disease, we're not going to cause a mutation. We're not going to have to use it in a prolonged period. It's going to be pulsed or whatever. So what I'm trying to say is be open to possibilities. We have to, this disease has been around for a long time. If we want a cure, we have to look beyond the obstacles. We have to look about how to go beyond those things. So when we, just remind you again, review each other's papers, let's be supportive of possibilities and not focus on too much of what might not work. Let's focus on what can overcome those problems.
Okay. Thank you, Duke. I think Ron, oh, you've got it. Go right ahead.
Hello, I'm Ron Cooper. I'm the CEO of Alagille Pharma. We're one of those small companies that's trying to advance a treatment, for cholestatic disease and diseases like, Alagille. And I think the first challenge, these are rare diseases. So what does that mean? That means that information is rare, right? So information on the disease, on the natural course of the disease, on insights and potential. And you talked a lot about a lot of the complications. There's a lot of heterogeneity between the patients as well. So that, by definition, makes it tough. So we are highly dependent upon the scientific community, the regulators, and on the parents and patients themselves. And how are we dependent upon them? On the scientific community, the things that we heard yesterday was absolutely phenomenal, right? Yesterday was a great day because it put together a lot of folks from a very high level of kind of state of play to how things are being treated to a lot of detail. But that doesn't happen very often, right? And that's a lot of bits of information that have come together so that when we have more insights on the different patient types, the natural history, what's really going on, it makes it a lot easier for us to advance drugs. That's one thing that we need. We need science. But we also need help from other individuals and the regulators. And in fact, many of you might be aware that there was an act called the Orphan Drug Act that was passed now a couple of decades ago. But that was transformational for companies like ourselves and other individuals to come into that space because it provided incentives, right? And unlike many of these researchers, companies like ours we don't sell anything, so we have no revenue. So we need to get grants, and our grants come from investors, right? So we have to be able to show potential investors that the research that we're doing makes sense, right? And that over time that will provide them a return. So that legislation is really important from multiple perspectives. So science, regulators, and the third part, yourselves. The insights are so important for us, right? Because we're gathering bits of information. So your experiences of what you've gone through the system, what happens with your children, your interest in scientific trials, what type of trials you would be involved in is so important to us. And we're really grateful when you participate in trials because we know that there's it's a big deal, and there's anxiety that's associated with that. And it's your one child, but that one child added up with another group of child helps us get some important answers, right? So, let me start where I began. These are rare diseases, and rare diseases means that there's not a lot of information around. When we can collect that scientifically with the regulators and with yourselves, it makes a big difference. And yesterday, Dr. Piccoli said, he's been in this in a long time. He said, I just can't believe the number of people in the room here today that are interested in Alagille syndrome. A couple of decades ago, that would never have been the case. And that's because of the advances in science, in the regulators, and in the participation of patients like yourselves. So we thank you for that.
Thank you, Ron. All right, Beth?
Hello, everybody. I am Beth Sloggy, and I am from Reprogramming, a small biotech company in San Diego. And I'm very happy to follow Ron right now because I truly believe that drug discovery is a team sport, especially to be successful. And in rare disease, we need the help of everybody. And I'm also very excited to be here and to have the members of the Reprogramming team, as well as our partners at NCATS and NIH, and to say that we have just recently entered a new partnership with the Alliance, a three-way partnership that is going to require the help of everybody in this room to be successful. We are entering a new area for us and aiming to take multiple shots on goal to see what we can do to target the underlying root of the disease and develop a transformative treatment. And we're not going to do it without the experts, the doctors, the families, physicians. And so I just want to also ask for everybody's kind of patience. I know we may ask questions that some people have already answered before, or that parents or families are very tired of always answering, but I promise you it's helpful and it's meaningful, and we are very excited and very humbled to have been so welcomed into the community thus far. So thank you also.
Thank you, Beth.
Hi, I just wanted to say good morning, and I'm Nancy Spinner. I'm a geneticist, and I've been working on Alagille syndrome since the very beginning. And I know there were several questions that were on the patient list of questions that had come up about genetics. Why do some people have mutations when neither parent had a mutation? And what can you do if you've had one child who has a mutation? What can you do for family planning? And rather than try to answer those here, tomorrow morning I'm giving a session on Alagille syndrome, genetically speaking, where I will attempt to answer all of those questions. If you have anything in particular that you want to make sure I cover, please come let me know today. Otherwise, we'll go through all of it tomorrow, and I'll have some slides to hopefully make it all pretty straightforward, and you can ask all of your questions. Thanks.
Okay. Thanks, Nancy. Anyone else? Oh, over here. Oh. We're working you today.
Hi, everyone. I'm Natalie Pfirrman from Discerna Pharmaceuticals in Cambridge, Mass. And I just wanted to introduce ourselves. We're new to this community. We're really happy to be here. I want to thank Stacy for bringing us in for the scientific meeting yesterday and for this today. I want to echo, what everyone else has said here. We need to learn from you. We're new to this field. We work in rare liver disease. That's what our company specializes in, but we're small. We can't be experts in all these diseases we're working on. We have experience with similar rare diseases, getting them into the clinic quickly. We're very interested in Alagille and learning more from all of you today and seeing, your experiences really guide where we're going to go with our treatments, how we design our clinical trials, what kind of side effects do we need to be concerned about, what are the other symptoms other than what we've seen in the publications? What do we need to look at? So we're really looking forward to learning from all of you and hopefully being here for many years to come to bring it forward. So thank you.
Thank you. Okay.
Hello, everyone. My name is Wei Zheng. I'm from NIH, and as Beth mentioned, we just started this new collaboration with with the foundation and retrofit for the drug development to treat this disease. So I want to give you a little bit introduction of our program, and we have this called the Therapeutic for Rare and Neglected Diseases program at NIH. So our mission is a translational research. It's a it's a work with the basic research scientists in academia, and also the biotech company, and together move the drug from the discovery through the preclinical discovery and with the FDA for IND into the clinical trial. So that's our mission. And in the last 10 years, we have worked with the biotech company and academic professors on over 30 projects. You can see in our website, NCATS, rare disease projects, and we have successfully moved over a dozen of the drug into clinical trial. So this is the first time I'm in this scientific meeting. It's a lot of things for me to learn, and I just feel and saw so much new advancement in this area, and it's a good starting point for us to work together to do the translational research to move the therapeutics for the patient. Thank you.
Thanks, Ray. Anyone else? Okay, I think we can go back. We can take maybe 2 questions if we want to go back to that format, if family has questions.
Hi, I'm Amy Foley. My daughter is Elizabeth Foley with Alagille syndrome. She's 2.5. I constantly hear about these trial drugs, and I just have no clue how to even reach out to anybody to figure out how do we get involved? How do we get our child involved, and where can I go to get more information or request to be part of that? Thank you.
Yeah. Okay. Yeah.
There are a couple of companies that have been working on drugs called ileal bile acid transport inhibitors. Specifically, those are probably the ones you're talking about, that are a novel way of treating itching. And the way that they work is that they inhibit a transporter in the bowel that brings bile acids back into the circulation, so then it allows the bile acids to go out in the stool. And we in the Children Network have done one study already in Alagille syndrome in collaboration with the Lumina Shire, now Miram group. And then I know that Albareo is also interested in getting a trial off the ground in this space as well. So I think for the moment, what I would say, and Ron can add to this too, but I think there are going to be a couple of new trials in planning in Alagille syndrome. And right now, we don't have anything new for enrollment, but I think we will soon, and you should kind of watch the space. Do you have anything to?
David?
And contact your doctor. There's also clinicaltrials.gov. Anything new that's ongoing, that's enrolling, is going to be listed. And you can put in Alagille syndrome on clinicaltrials.gov, and that would be a good place to start in terms of knowing which centers are involved with all these trials. And then how you might contact. And then there'll be a contact person on there that you could reach out to. And hopefully there would be something nearby where you live. And even if it's not nearby where you live, sometimes there's ways to, some of the trials will pay for families to travel to places that they could go for the study visits and still maintain their care at their home center, and then go be seen at a center that's participating in a trial and be part of the study visits that way. So that's, and then the Alliance may have some information on your website if you know of new trials that are starting up that are enrolling patients with Alagille syndrome.
Absolutely.
So those are all good places to look.
Could you just say one word about the Children Network? It has nothing to do with Children's Hospital.
Yes.
Yeah.
Children Network is the Childhood Liver Disease Research Network, which is an NIH-funded consortium of 13 centers, and our center, Children's Hospital Philadelphia, as well as University of Toronto, Sick Kids are 2 of the centers that are part of it. And we have ongoing enrollment of kids with Alagille syndrome. It's one of the many diseases that we study within the Children Network. So we have enrollment at all the children's centers for patients with Alagille syndrome to be followed long-term. We have these longitudinal studies that are mainly just observations of clinical information, having a visit once a year, getting all the clinical data into our databases. There's some collection of DNA and plasma and serum along with that. So those are kind of our ongoing studies. And then now, hopefully in the next few years, children may be starting some new clinical trials as well, which are still in the planning stages. And again, if any of those are starting, information would be put on clinicaltrials.gov and would be available for people to reach out and potentially participate.
Thanks, Kathy. Okay.
Hello, my name is Jennifer Aponte. I'm from Florida. I have a 7-year-old warrior, and I attended the scientific meeting yesterday, and it was very interesting to hear all of the information that we got about vascularity and how notch affects vascular development and this and that. I wondered if there is any data to date about what clinically is significant, what traits we need to look for in our children. Is there anything that we need to watch out for? Is there an age to have like a brain MRI or a vascular study, any kind of flag that would tell us, or is this something that we could do preemptively? So I guess it's a question for both the clinicians and the researchers.
Okay, great. So there's still lots of big gaps and things that we don't know. So one recommendation would be that if your child had a significant head injury, that they should definitely have a more aggressive evaluation in an emergency room than would be by standard emergency room guidelines. So that would be one issue. If your child had any, if your warrior had any new symptoms that were concerning, weakness in an arm or leg, slurring their speech, then that would go straight to emergency room and get an urgent evaluation. I think those two things are fairly straightforward. From a screening standpoint, that means doing something if there aren't any clear symptoms, getting a scan. That's a little bit harder. We don't know for sure. The recommendation that we would have is that if your child is having any major surgical procedure, could be any type of surgical procedure, you should probably get an MRI of the head before that, because it might be important for the anesthesiologist to know about something like that, because blood pressure can shift while you are under anesthetic. And we also do that routinely before liver transplant as well. That obviously counts as a major surgical procedure. All of that put aside, if there's none of those things are happening, no head injury, no symptoms, no surgical procedure, we would do a baseline MRI of the head, typically around the age where you don't need sedation. So that's about 8. And that's it. What we don't really know is what to do if the scan is normal, as to how frequently it should be followed up. I would say roughly every 3 to 5 years after that. But that's a personal recommendation. We don't really know. It's not data-driven. But I think there should at least be a baseline MRI of the head. I would also advocate to the adults in the room who are affected that you should also get a baseline MRI of the head, because even though you may not have been very affected by Alagille syndrome from a liver disease standpoint, we don't know about, really know a lot about the frequency of blood vessel abnormalities in adults. So I think that risk is there. And then that was all about the head. In the abdomen, we know that there can be blood vessel abnormalities in the abdomen as well. And I think primarily, if no one's having any symptoms, we wouldn't do a routine scan, except if you're coming up to for liver transplant, because it can be relevant in planning aspects of your transplant.
Okay, thanks. One last quick question before we move on. Sure.
Yeah, Chris Peets from Miram.
Chris.
Just looking to follow up on the clinical trial question from wherever it came from. And so 2 points. One is tomorrow there's a session where Pam Vig, our Chief Scientific Officer, is going to talk about clinical studies in the space and a bit about our program, Maralixabat. And also from our planning standpoint, we're working with FDA to get Maralixabat available for Alagille syndrome patients as quickly as possible. And are planning to have another study open next year.
Okay, great. Thank you. All right, one more.
I just wanted to say that I've worked with other alliances and networks, in particular lymphedema, and a lot of the good things that have happened in terms of what can be done is educating the NIH, CDC, or any other government agencies about the importance of your particular syndrome. So the NIH website, nih.gov, used to be a little tricky to find information about different diseases, and it's improved tremendously. The NIH is your agency. It's taxpayer-supported agency. It doesn't belong to anybody else other than the United States citizens. And if you look up on their website, I just looked right now, there's a whole section on treatment of Alagille syndrome from NIDDK. There's a section about clinical trials in Alagille syndrome. So I think it's important to be able to have one-stop shopping for, people who want to find information that's authoritative. And if it's not good enough on the NIH.gov website, that the Alliance may want to interact directly with them to say, these are the problems we see. That's exactly what happened with the lymphedema patient network. CDC had nothing about lymphedema, and 10 million patients have serious infections. So going directly to the agencies and asking or demanding for improvements will allow you to maybe have your pediatrician say, oh, please go to this NIH website to learn about Alagille syndrome. So I just wanted to throw that in there.
Great, thank you. Okay, we're going to break into our working groups, but I saved us a couple of extra minutes because this first time is going to be a little bit confusing. If the moderators would stand and let me move them maybe here and there. Shami, if you can move over here to this one, and Shanda or Sandra, either one, if you could move maybe to that next table. Yeah. Where is that? Okay, yep, you're right there. Okay, so Jeff, can I have that first question slide? Perfect. And let's turn off well, actually, can I have one? Okay, so, all right, we have 8 moderators, and we're going to take one question at a time, and what we were going to do is go through these questions, and you choose which table interests you the most, and that would be your first working group. So since you don't know all the moderators, let me introduce them really quickly, and then they're going to hold up a number, and you just kind of follow that number. And 2 different tables will have the same question. So Stephanie Davis, who we heard from earlier, is one of our moderators, and Hannah Craig, who we heard from earlier. In the back is Sandra Beltran, who just arrived from Spain, and she's one of our moderators. Shanda Turner is also an Alagille mom. Tara Jock is not only a mom, but also on our board of directors. Jen Aponte, in the very back, is also an Alagille syndrome mom. I think that's it. Okay, so question number 1. Oh, Shami. Oh my goodness, Shami. Shami is also on the board, and Shami traveled for 4.5 days to get here from India. Sorry about that, Shami. Okay, so question number 1. If you're a number 1 moderator, if you'll hold up your card. Oh, Stacy. Oh my goodness. All right, Stacy also, Dr. Huppert is a moderator. Y'all keep me on track. All right, question number 1. 40% of the survey respondents were misdiagnosed. What can prevent this and lead to quicker diagnosis? Is it more than education? Is genetic testing an answer? How can this percentage be drastically lowered in the coming years? That's question number 1. Number 2, 85% of survey respondents take medication. However, 43% say the medication is not effectively treating their symptoms. Is variability the only cause that the medications are not being effective? How can this be addressed in the clinical setting to improve the medication outcome? Number 3, 42% of survey respondents said their physician was not knowledgeable about Alagille syndrome. What are concrete ways the knowledge gap can be addressed and drastically lower this percentage in the next few years? Number 4, 62% of survey respondents have received at least one insurance denial. Can families coming together along with their clinicians and patient advocacy groups impact this? What type of systems could potentially be put in place to change this high rate of denials? So if you guys would hold up your numbers. So number 1 will be Tara and Dr. Huppert. Number 2 is Shami and Jen in the back. Number 3 is Hannah and Sandra. And number 4 is Stephanie over here and Shanda. Where did Shanda end up? There, Shanda right there. So we're going to make our way to their tables. You can leave your stuff at your table except maybe a pen and paper. And once we get there, situated at our tables, I told you, musical chairs, then I'll tell you about the goals that you'll have. Okay, yeah, so you'll go to the table with the question that is most interesting or impactful to you. Hold your numbers up high. And we need to get a good mix at each table of families and scientists, clinicians, industry folks. So let's make sure we have a good mix at each table. And once you get to the table that you want to be at, please just go ahead and take a seat. If you need pen and paper, there are some pads and some pens at the back of the room on the water table. Two. Two is right up front here. Okay. We need a family up here. Oh, there you go. So let's see what question. What's the other number 1 table? Tara. Okay. Need a clinician? Are there 2? Okay. All right. Yeah. Okay, that went well, guys. Thank you. Wasn't sure how that was going to go. Okay, so keep the question in mind. Your moderator is going to take notes, and at the end of the day, you will have 1 hour on this question, and then we're going to break for lunch, come back and have one more question, which we'll have an hour for. At the end of that, your moderator will come up here, and anyone from the table is welcome to come up as well and present kind of what you talked about, what your findings were, and what our roadmap is. And here's our goals. So keep these in mind, and each one of your moderators has this. So goal number 1, which I've kind of done a little bit of this already just from the survey. We want to identify gaps in shared knowledge and priorities. What are the true challenges associated with the proposed question? Okay, and then goal number 2, develop a list of potential solutions in P-course studies, keeping in mind our overall goal will be to walk away with a roadmap to inform future scientific inquiry and therapeutic discovery and positively impact patient care. So we're going to come up with what the actual challenges are, and then let's try to come up with some solutions. And we will have 1 hour. I'm just going to be walking around. Roberta, I think, is going to be walking around. And possibly, what did I do? And Dr. McGee also will be walking around if there are any questions about how to facilitate. Okay, go ahead.